Welcome to Ajit Bioacademy. Today I will talk about neurotransmitter release mechanism. How neurotransmitter release from the synaptic vesicle towards the synaptic cleft, and how it will fuse the terminal membrane. Okay, so I will discuss in molecular level. So before going to the details, I will just request you to watch my previous videos: action potential, neurotransmitter synthesis, neurotransmitter loading into the synaptic vesicle. So it will be very easy to understand because those terminology will come here, are connected here. Okay, so now today's topic is neurotransmitter release. Neurotransmitter release. Okay, so before going to the details, I want to give you some basic idea about this, and then I will go for details. So this is a presynaptic terminal, and this is the post one, and this is the synapse, this is synaptic vesicle. Okay, and this is the synaptic cleft. So here the synaptic postsynaptic membrane carry some corresponding neurotransmitter receptor. So if you think this is the dopaminergic neuron, so here dopamine. So when the action potential will reach basically depolarization current, and then by the influence of that depolarization current, calcium channel will open, and calcium will enter and fuse the vesicle, and neurotransmitter will release, and that neurotransmitter will bind the corresponding receptor, and it will activate the post synaptic neuron, or you can say target effector organ. Okay, so the question is, this is the very rough idea. In generally, in different books, as just written in that way, but actually, in molecular level, how exocytosis will happen. So today's topic is neurotransmitter release in molecular level. So now, for go for details. So before discussing, for your easy understanding, I will categorize it in 4 categories. One is tethering, second one is docking, and third one is priming, and fourth one is fusion. Okay, so just roughly I want to just introduce you with this terminology. Tethering means basically this is loose connection, very just come to close to each other but not connected. Okay, and docking means it's attachment. It will attach but not very closely attached, not form any complex or interaction. And priming means is ready to use, ready to go. That means before final round, just final preparation. That is the priming. And fusion means the final stage. Okay, so if you try to just park your car in a particular parking areas, so at first you have to just come close to your parking area, then you have to enter in your parking zone, then you have to just adjust your car to the proper positioning, and finally you will fix it. So now uh, going to the details, today's topic. So tethering and docking. Tethering can be considered as a loose docking, or somebody says it is pre-docking, or you can say loose uh, loose binding or loose connection. Okay, so sometimes in 2 points in a row you can say this is docking. Okay, but for better understanding, I will say tethering, docking, priming, and fusion. And by this way, neurotransmitter will release properly. How? Now come to the details. So before going to the details, I will draw a basic structure of the vesicle, and then basic structure of the membrane, and then we will try to make the connection to each other. So here here is the neurotransmitter vesicle. Membrane is basically a membrane, and it carry basically an alpha helical structure that is, you can say, synaptobrevin, as it is attached with the vesicle, and inside that these are the neurotransmitter. So these are the neurotransmitter. Okay, so you can say its another name is B snare, as it is attached with the vesicle. So vesicular snare, that is B snare. That is synaptobrevin. Also attach here a synaptotagmin. Synaptotagmin. Okay, and you can say this is a calcium sensor. Okay, and here is two domain, the calcium binding domain, this one and this one. See, 2A and 2B, this is calcium binding domain. And also here another things is, this is a protein, attached protein, and this is also transmembrane. This is also transmembrane. Okay, and this is the attached protein. This is called RAB protein. Okay, a RAB protein. You can say RAB3, RAB27. These are RAB protein. And RAB protein initially attached with a GDP, inactive position. It is attached with GDP. Okay, that is initial stage, inactive stage. And besides this, another attached protein is there, come from another site, attached with the membrane, the pre-attachment, and this protein name is, that is guanine nucleotide exchange factor. Okay, so this is the GEF, which is initially attached with the membrane. So initially, this is a very much important molecule attached with the vesicle, synaptic vesicle, which are directly connected with the tethering, docking, priming, and fusion. Okay, and for your easy understanding, now I will draw membrane, that is plasma membrane. You can say presynaptic terminal. This is lipid bilayer, and here something is connected. One is, here is this one is, I can draw how. For your easy understanding, I'm just drawing it far away, but here is very attached, because when the release procedure will start, actually the vesicle is very adjacent to the active zone. Okay, this is ready or matured synaptic vesicle. So that will be very adjacent. Then you can say radipole, and just behind the radipole vesicle, another vesicle will wait for next step, that is the reserve pole. So for understanding, I'm drawing the very far distance, but it is very attached. So now this is the calcium channel, and one protrudes part of calcium channel here, and this calcium channel is directly attached with a particular protein that is called a rim protein. Rim means that is Rab interchanging molecule. You can say it is the effector protein of Rab protein. So this is the Rab protein and this is the rim protein. Okay. So here another protein is attached, that is I can just attach here. This is the Rab binding protein. Okay, that is also attached with the calcium channel protein and rim protein. Okay, so this is the initial stage before activation. Just they are attached very adjacent to the active zone, and no signal is not attached here. That means nerve is in active stress. So when nerve will get signal, the neurotransmitter vesicle, that is synaptic vesicle, will get signal, and it will be already loaded and matured, which is just remaining adjacent to the active zone. When it will be activated, get by actin, cytoskeleton, or other molecules, then the GEF protein will be activated. That means guanine nucleotide exchanger protein that is attached with the membrane, it will be activated. And then what will happen? It will activate the Rab protein. How it will activate? It will activate as it is named is guanine nucleotide exchanger factor. So it will exchange the guanine. So how? It will exchange the guanine to in which color I can use? So GDP to GTP. So here is it will just go out or detach from here. So this is the GDP, and here the protein will come, that is the GTP. That means here GDP will exchange to GTP by the activation of GEF. This is exchange, not conversion, as like G protein. So you know G protein is the different subunits are there, alpha, beta, gamma subunit, hexamer. So here is only monomeric Rab protein. Rab protein is monomeric protein. So you can say Rab protein is called, you can say, monomeric G protein. Okay, so when GEF will be activated, that means this vesicle will be matured and will get the message. Then GEF will be activated, and that will activate the RAB protein. Because when GDP will exchange to GTP, then it will change the conformation of RAB protein, and RAB protein will be activated. And what will happen? This RAB protein will attach with the RIM protein. Okay, directly it will attach the RIM protein. And this, when the RIM protein will bind with the just RAB protein will bind with the RIM protein, then the RAB binding protein will make this bonding is very stable. Okay, and here other substances, other scaffolding protein are very much rich in that area, that is active zone. Already you can consider it active zone. Here the cyclometrics are very rich, different types of actin, cycloskeleton protein, different scaffolding proteins. You know, the RIM protein you can consider as a scaffolding protein. Here some other supporting scaffolding proteins are there. Then I can just write it down here. That is, you can say piccolo. You can say that is the basin protein. You can say CASK protein. So these are the very basic scaffolding proteins, or just a movable protein in the active zone that will make this conversion in or conversion very stable, and it will direct the vesicle towards the membrane in right orientation and proximity. Okay, basically this basin protein will organize the event, and this will contribute for the stability and stability and their other conversion. But basin protein will organize the event. Okay, so firstly, what is happening? That is, when GEF will be activated, then RAB protein will be activated by exchanging GDP to GTP. And activated RAB protein, that is, you can say RAB3, RAB27, will bind with the RIM protein. And RIM protein, you can say this is the effector protein or RAB protein. That is RAB effector protein. Okay. You can say this is also a scaffolding protein which is directly connected with the calcium channel. Okay, and also connected with the RAB binding protein. So, sorry, here I did a great mistake. That is the RIM binding protein. I'm so sorry. RIM binding protein. Okay. Now this event done. So this is the event is tethering. No connection with other molecules or factors with the membrane. Just they are making connection with the scaffolding protein and calcium channel. Okay. Now we are going to details the next step. Now I have to draw the plasma membrane proteins, some plasma membrane snare proteins. So the snare proteins are basically one is this is alpha helical transmembrane. Okay, transmembrane. And this protein name is syntaxin 1 basically. So this is the target membrane. So you can say this is the T snare. Okay. Here another protein is there. There is double helix, very attached protein by lipid, This is helical. This is helical. Double alpha helical structure. This protein, attached protein, the name is, that is SNAP25. Okay, this is also TSNAP, the target SNAP protein. This is the membrane, plasma membrane attached SNAP protein, and this synaptobrevin is attached with the vesicle. So this is the B SNAP. So this is the SNAP protein. These are the SNAP proteins. Both all are alpha. This is single alpha helical structure. This is single alpha helical structure, and this is double alpha helical structure. And in the initial stage, in syntaxin, each is just folded its motif within itself, and it is covered by a protein. This is called Monk 18. This is called Monk 18. Already I've forgotten one protein is also some gives some contribution, that is which color I can apply, mixed color I can apply here. Oh, this is just like roughly I will give you a signal, that is the Monk 13. It will play an important role in the priming stress here. So this is the Monk 18 is attached in the active site of the syntaxin, and this syntaxin is now inactive. Okay, so now what will happen? The next step is docking. The next step is docking. So docking means, already I said, you can say attachment. So here some attachment will occur. So initial loose attachment is just between the Rab protein and scaffolding protein, rim protein. This is loose connection, and the proximity and orientation will be maintained by different types of supporting proteins, and that will turn into very close to the snare, V snare to the T snare. Okay, so now I will just draw this vesicle to here for your easy understanding. Okay, so now this is your vesicle. This is your vesicle, and inside the vesicle, this is neurotransmitter, and the snare protein will be V snare will be here. Okay, just adjacent to that snare protein, but these are not connected. And already you know here also carry the synaptotagmin. So this is just changing the position and orientation, and it is just very adjacent, or you can say attach. You can say attach with this protein, but not making any bond or make any complex or cross linking. Okay, just proximity and orientation. You can say it will be confirmed by this stress. That means the Basson protein and piccolo protein and other Monk protein will contribute there, and it will just change the conformation, or you can say change the position of the vesicle, and it will come to close to the towards the membrane position, and the snare proteins, the base snare and T snare, will come to close, very close to each other. Okay, but not directly attached like cross-linking. So I will say here during tethering, the vesicle and membrane distance is 25 to 30 nanometer, and during docking it's 5 to 10 nanometer, which is very adjacent. Okay, so this condition is called docking. This is docking. So docking means attachment. That means here one attachment between the RAB protein and the rim protein, that is scaffolding protein, is attached with the rim protein, directly connected, attached, and the orientation has been changed by this different types of factors and proteins, and then the vesicle is just come towards the B snare to T snare, and B snare T snare is just positioning very adjacent, you can say roughly attached. So this is called docking. Okay, this is the docking. So now what will happen? Now go for the third stage, that is for your priming. So during priming, what will happen? These three snare proteins, one single helical synaptobrevin, double helical SNAP25, and one helix, that is syntaxin, will make a cross complex, that is like G-peri. Like these are just positioning in that way. Okay, and this is a slider, and when it will just you will just pull upward, then it will close as like a zipper. So they will just make a zippering, and this is the priming. So it will, how it will happen? So now here one protein will arrive here, that is a protein is called MONK13. So here I just draw in different color. So here I can draw this in red color and blue color. So this is your MONK13 protein. So then MONK13 protein will displace this MONK18. Displace does not mean the detach. It will not detach, that displace. It will displace from active zone to the nearby position. So that will your MONK protein will be there. Okay, that means it will be displaced by the MONK protein, MONK13. MONK13 will displace MONK18 from the active site. Okay, it will open the motif of the syntaxin. Okay, that syntaxin is now open. Okay, then what will happen? It will make cross-link with other snare protein. Okay, so what will happen? So in the beginning of this priming stage, MONK13 will displace the MONK18, and it will open the syntaxin, and syntaxin is ready to just make a complex with other snare protein. So what are the snare proteins? The single alpha helical synaptobrevin, double helical SNAP25, and itself the single alpha helical syntaxin. And it will come to close each other, and it will make a complex by cross complex and like G-pairing. Okay, so I will just draw in a separate way here, and you can just you can just show the closure of the vesicle. So now I will draw, that is your vesicle, that is your vesicle, and this is synaptobrevin, this is your synap25, and this is your syntaxin, and these are already make a cross complex, that is the cross complex, and you can say like G-pairing like that. So you can say this is the G-pairing. Okay, so if it is happening in that way, then what will happen? Without coming signal to the action potential, signal of calcium action potential, it can start its fusion. So premature fusion may occur. So instantly the priming procedure will complete? No. So here one protein will just attach with this G-pairing complex or cross complex protein. That is called, you can say, this is a this is called complexin protein. Okay, this is the complexin protein. So what is the function of complexin protein? Complexin protein will just prevent the whole G-pairing, the complete G-pairing. It will ensure the initial G-pairing, partial G-pairing. So what will happen now? Now complexin is attached here, and it will just stable the G-pairing, and it will prevent the final G-pairing, and it will ensure the partial G-pairing. Okay, so before going to the next step, that is fusion, so another things will happen, that is in that stage the RAP GTP attached there, the RAP GTP will hydrolyzed by gap protein. That means GAP protein will just hydrolyze the GTP, which is attached with the RAP, and GTP will convert into GDP, and this connection will be loosened. Okay, and this complex formation, cross connection, or you can say 4 alpha helical complex. 4 means your one helix, one alpha helix is from that is synaptobrevin, that is B snare. One is you can say syntaxin, that is T snare, one alpha helix T snare, syntaxin, and 2 double alpha helical structure is SNAP25. So that is the 4 alpha helical complex. This 4 alpha helical complex will be more close to each other, and it will form a alpha helical complex, and suddenly complexin will come and it will make a stop for final G-pairing to ensure the partial G-pairing, and it will wait for the next step. This step is called priming. So priming means now this condition is ready to fuse, ready to take part in fusion process. Okay, so this is the priming. So in this priming stage, actually the monk 18 will displace by the monk 13, and conformational change happening, and the snare complex will form. Snare complex will form by the synaptobrevin from the V snare, that is vesicular snare, and T snare, that is syntaxin and snare 25. It will make a 4 alpha helical complex and partial gibbering. Okay, this is the priming. And now I will come to the fourth stage, that is the fusion. So what will happen? The fusion. So here I will just show you the fusion. So what will happen? So I will just displace this vesicle, like draw this vesicle here, like, okay, and here it will carry already, you know, this is your synaptotagmin, and two domain, calcium binding domain is that one and this one, that is C2A and 2B, this is the calcium binding domain, and synaptotagmin is calcium sensor protein. Okay, so now the fusion. So now what will happen? When your nerve will be excited, stimulated, then action potential will create, and then depolarization current reaches to the terminal. Okay, this is membrane, that is the active zone. Depolarization current, and depolarization current will be produced by the entry of sodium. And when the depolarization current will reach, by the help of depolarization current, the calcium channel will open. So this calcium channel is voltage gated calcium channel. That means the channel will open by the influence of voltage. So what is the source of voltage here? This is the depolarization current. So when the voltage will arrive here, depolarization current, it will open, and this here calcium will enter. So you can say these are the calcium, and calcium will here. So already I changed the position, so that is somewhat a little bit confusing here. So don't get wrong. Just this is the calcium. So this calcium will bind here. Okay, and this calcium will change the conformation of this synaptotagmin calcium. And then what will happen? When calcium will bind with the synaptotagmin, that is calcium sensor protein, which is attached with the vesicle as a cross membrane, like transmembrane. So now what will happen? It will change the conformation. After binding the calcium, the synaptotagmin changes conformation, and it will bind with the complex. How? In that time, by the influence of calcium binding to the synaptotagmin, the calcium will influence this condition and change the conformation. And what will happen? The complexin protein will detach from here. Okay, this complexin will out. So no complexin here. So complexin will detach, not displaced, that is detach. And here, so this will bind with that. So it will bind with the cross complex, that is alpha 4 alpha helical complex, will just influenced by the synaptotagmin, and it will create a mechanical force. So basically mechanical force will be created by the cross linking of the snare proteins and the calcium, influence of calcium binding synaptotagmin. And by the influence of mechanical force, this membrane will fuse that membrane. Okay, this is the membrane of the vesicle, and this is the membrane of your presynaptic terminal, and that will fuse. I will draw in that way. Okay, and here the neurotransmitter will come out by the exocytosis process. Okay, and this neurotransmitter then finally stay in the synaptic cleft, and it will bind with the postsynaptic membrane receptor. Okay, and beside this is the periactive zone. Over there it will be recycling. I will not go there. So this is the fusion. So during fusion, what will happen? Actually, during fusion, the complexin protein will be detached by the synaptotagmin, charged synaptotagmin, which is already bind with the calcium, and finally it will make the total conformational change of this complex, and it will create a mechanical force, and that will fuse the membrane, vesicular membrane to plasma membrane, and then it will just release the neurotransmitter by exocytosis process. Okay, and in that time, here another things, NSF and SNFP will make a complex. This SNF has a active ATPase activity, and it will detach the snare complex, and it will again recycle. So recycling is different part, so I'll not go for this today. So this is all about the neurotransmitter release. So this is the tethering, means just loose attachment. You can say pre-docking. Here just different types of scaffolding protein will contribute there. Basically reactive RAB, that is RAB GTP, will bind with the RIM protein, and here the RIM binding protein and MUNC13 and other scaffolding protein, that is picolinobactam and CSK, this will contribute for their proper conformational change, orientation change, and it will just move toward the snare proteins of the plasma membrane. Okay, and docking, this will ensure the pre-attachment of snare proteins, that is B snare and T snare. It will just come very close to each other. You can say it is already attached, but not making any conformational change or making any cross-linking, making any jippering. This is the docking. And priming means it will just make a 4 alpha helical complex cross connection, jippering, and here finally then fusion by the synaptotagmin or calcium channel. So here action potential is very much important, voltage gated calcium channel is very much important, and finally snare proteins. So snare protein B snare and T snare. This T snare means target snare, that is syntaxin and SNAP25. B snare means synaptobravin. They will make a bond to each other, that is snare complex. This is the main you can say complex for the fusion. And finally the synaptotagmin, that is calcium sensor protein, that will make sure for conformational change and producing a mechanical force for the final fusion, and that will completely depend on calcium, and calcium channel will open by the influence of action potential, and action potential will generate after activation of the nerve or stimulation of the nerve at the level of threshold or more. Okay, so this is a series of events, that is tethering, docking, priming, and fusion. So already I discussed in molecular level. So if you have any questions, then you can comment, write in the comment box. And if you think this is the what you want to share, so you can share with your friends and the knowledge seekers. So thank you very much.